Clinical trial endpoints: primary versus secondary versus exploratory versus sensitivity versus multiplicity. Introduction. In a clinical trial study, endpoints are the measurable outcomes used to evaluate whether a treatment is effective, safe, or clinically meaningful. Different types of endpoint analysis serve different scientific, regulatory, and business purposes. Clinical trials made easy: primary endpoint analysis, the most important outcome that decides study success. Here the definition of primary endpoint analysis is: the main outcome of the study is designed to evaluate. It answers the most important clinical question. Regulatory approval decisions are primarily based on this endpoint. Now the key characteristics for primary endpoint is: predefined before study starts, derives sample size calculation, usually only one or a few primary endpoints, highest regulatory importance, and controls study success and failure. Now the real world example here I would like to explain here is for the oncology. Say the study objective is evaluate whether a new immunotherapy improves survival in lung cancer patients. So the first primary endpoint is the overall survival, OS. This is the main endpoint in any clinical trial study. So the analysis here is to compare treatment versus placebo using Kaplan-Meier survival curves, then Cox proportional hazard models, then hazard ratio HR, and the log rank test. So these are the analysis and these are the specific endpoint. Now say for example, if the hazard ratio is 0.72, that means the treatment reduced death risk by 28%. That is 100 minus 72%, which comes to 28%. And here the p-value is significant, 0.003, which is less than 0.05. Typical statistical methods that are used in primary endpoint analysis are: say for example, if the primary endpoint type is continuous variable, so in those especially common analysis is used ANCOVA or mixed models. Now if the primary endpoint variable type is binary, then the logistic regression is used. Binary is nothing but 0 and 1. Now time to event, if it is related to success and failure or survival or death, or it's related to censor and uncensored, whether it's related to event and no event kind of category, kind of a data, which you will mostly see in the oncology clinical trial study. So in that specific primary endpoint time to event, you will generally see Cox model, log rank, then Kaplan-Meier curves, and all these kind of analysis. Now, if the primary endpoint type variable is related to count, right, counting the numbers. So in those you can use Poisson or negative binomial. Secondary endpoint analysis, the key purposes here is to support primary endpoint findings, provide supplementary information, explore additional clinical benefits, and generate hypothesis. Now primary endpoint analysis is the first kind of a main outcome or the endpoint, right? After that comes the secondary endpoint, which will give the additional clinical benefits to the clinical trial for exploring more kind of parameters. So here the definition is: secondary endpoints evaluate additional effects of treatment beyond the primary objective. So the primary objective is there. Beyond this primary objective, if there are any additional effects that can be seen to evaluate the treatment, so in those scenarios, those secondary endpoints are analyzed. Now these study help understand safety, additional efficacy benefits, quality of life, and mechanism of action. Now here the real world example is a diabetes trial. Now say for example, if the primary endpoint is HbA1c reduction. Now what happens in the diabetes? In diabetes we want to see the glucose level, whether it has reduced or not. So here the primary endpoint is HbA1c. Now the secondary endpoints with respect to this HbA1c reduction is nothing but the weight loss. Now if the treatment of diabetes clinical trial, that molecule or medicine is taken. So after consuming that treatment, whether the weight has reduced, whether the fasting glucose value has increased or decreased, or whether the blood pressure has decreased or increased, whether it's a low blood pressure or a high blood pressure, or it's a normal blood pressure. So these are all the additional effects, the endpoints that can be seen and that can be analyzed. Then hypoglycemia events. Now, if the secondary endpoint shows significance, study may still fail if the primary endpoint fails. Now, understand one thing: in any clinical trial, be it any therapeutic area, primary endpoint are the main focus and the main outcome. If the primary endpoint analysis and the interpretation fails in any clinical trial, no matter even if the secondary endpoints they show significance, the entire clinical study fails. So what happens in such scenario? Regulators prioritize primary endpoint. So primary endpoint is the main picture, especially in the clinical trial study. Now the statistical consideration here are: the secondary endpoints may require hierarchical testing, then multiplicity adjustment, which I will discuss in the later videos, then gatekeeping procedures to avoid inflated false positives. Now I will create another video in detail as to what is false positives with related examples. That specific terminology I will cover in the next video. Now third is the exploratory endpoint analysis. Now here the exploratory is nothing but to explore more apart from primary, which is main, and the additional benefits. So here what happens is, whatever that are post ad hoc measures that are used to explore the potential treatment effects and generate new insights for the future studies. So say for example, if I want to conduct the future clinical trial study, so to get the clear picture for conducting that future study, if I want to explore the current study and explore some other kind of endpoint, so that I can visualize. So what happens is, here the exploration of new biological or clinical questions that can be seen, or identify any potential signals, then generate hypothesis for future trials, right? Then support decision making and innovation. So here the key considerations are: hypothesis generating, interpret with caution and not for regulatory decisions, and plan and document clearly. Now here the definition is: exploratory endpoints are used for hypothesis generation, future research, biomarker discovery, and the subgroup exploration. Now these are generally not confirmatory. Now say for example, I would like to explain here, suppose I have an HIV AIDS kind of disease, right? Now if I have this multiple kind of a patients, now HIV AIDS can be seen even in the younger children, or it can be seen in the any kind of an age group. It could be the working age group, or it could be the older kind of an age group. So if I want to see the treatment effect across these subgroups of age, like say around 10 to 20, and then 20 to 35, then 35 to 45, then 45 to 65. So I have 4 to 5 different kind of subgroups, right? So if I want to see what is the impact of analysis across these subgroups, especially in the HIV, whether in which kind of an age group the treatment is more effective and it has benefited. So these are just kind of an exploration to dig into more kind of an analysis, and these are generally not confirmatory. So you can either add it or you cannot add it. So it depends on what the sponsor has recommended, and depends on what is the business requirement based on the costing. Now here the real world example is oncology. Now exploratory endpoints, it could be PD-L1 biomarker response, or genetic mutation subgroup efficacy. So like say for example, if I have only the efficacy which I have analyzed, but if I want to explore more the efficacy parameter based on different subgroups, it could be the ethnic kind of group or race, and it could be either the age group, or it could be some other parameter, right? Then immunoresponse markers. The example interpretation here is: we observed improved response among EGFR mutated patients. So this suggests future targeted trials. Now, if you see this kind of an interpretation that we observed improved response from this among EGFR mutated patients, so this is actually suggesting you to get the future trials that you can explore more in detail. Number 4 is a sensitivity endpoint analysis, where it is used to test, validate and ensure robustness. Now here the key purpose is related to evaluating robustness of primary endpoint, then assess the impact of different assumptions, support credibility and regulate confidence. Now identify potential biases and model dependencies. Now the sensitivity analysis is nothing but about the changes in assumptions, methods or missing handling data. The best example in sensitivity is replacing the missing data with some other imputation methods and see whether how it has affected the data and how it is giving you the different results or it's remaining same. So this helps confirm the reliability of the study conclusions. Now, the definition here is sensitivity analysis evaluates whether primary results remain consistent under different assumptions or analytical methods. It checks robustness, reliability and bias impact. Now, say for example, I have a missing data and a non-missing data. I want to usually compare the results with missing data and non-missing data. You see the analysis difference, whether it's remaining consistent or it is different. The another third part, it could be like replacing the missing values of the primary endpoint is respect to your last observation carried forward or the worst observation carried forward, or replacing by other means like say replacing by average or replacing by mean or median. So any imputation methods that you have used for the missing data, so that kind of analysis is one example that you can falls under the sensitivity analysis, and you want to check whether the primary results have been remained consistent or it is changing. If it is changing, then the clinical trial will go in some other direction. It depends on what therapeutic area, data and the primary objective is for that specific study. Now, why it is important? Now, clinical trial data often contain missing data, protocol deviations, dropouts, COVID disruptions, and non-compliance. So, these sensitivity analysis ensures conclusions remain stable, irrespective of including or excluding missing data or replacing the missing data by the imputation methods. For example, LOCF or worst observation carried forward. Now, even you can do the analysis by protocol deviations. Now, some people, they do it in different way, like say the protocol deviations are divided into two parts, minor deviation and major deviations. So say I have 100 patients, out of that around 20 to 25 are having protocol deviations, and out of that majority are related to major and minor. So what happens in such scenario, you want to generate two different types of analysis, having those patients which are having minor protocol deviations and those analysis with major protocol deviations. So in some therapeutic areas and studies, you will see this kind of table listings and figures being generated. Now, the real world example is primary analysis is missing data handling using MMRM, which is mixed model repeated measures. Now, the sensitivity analysis, like I said earlier, reanalyze using multiple imputation, LOCF, worst case imputation, and the per protocol population. Now, this is another one factor, population flags. Now we designed safety flags, ITT flag, MITT flag, then per protocol flag, kind of different populations. Now the sensitivity analysis is also one part, like say the first batch or the final statistical analysis plan, it has mentioned only safety and ITT population, and there is no per protocol kind of generation of table listings and figures. Now that set of table listings had been submitted to the sponsor. Now sponsor wants to see different kind of ad hoc kind of request. They have requested whether to include only per protocol population, and they want to see whether the results are showing significance or insignificance. So say for example, if the per protocol population or doing the ad hoc request, the results are showing significance, then it matters a lot. Then what happens, the clinical trial direction or the interpretation, whether it needs to be approved or it should go to the next level of submission to FDA, that direction itself completely changes. So it's very important, like when you work on the sensitivity analysis. Now if the conclusions remain similar, then results are robust. Common sensitivity analysis is related to, say, scenario is missing data, you do multiple imputation. Protocol deviations you have, so you include only those patients which have completed the entire study, which is per protocol analysis. Outliers is related to robust models, where you are removing the outliers and you are doing the analysis. And the COVID impact, which is pandemic sensitivity analysis. Now the regulatory importance here is regulators carefully inspect whether the conclusions change dramatically or they remain stable across assumptions. So if they change dramatically, then it gives the different direction to the clinical trial study. Now large inconsistencies may weaken approval confidence. Number 5 is multiplicity endpoint analysis. Now in this analysis, it arises when multiple endpoints or hypotheses are tested in the same study. It increases the chances of the false positive results, whether it needs related to control, adjust and conclude confidently. Now the key challenges here are inflated type 1 error rate, complex interpretation and regulatory scrutiny and justification. Now the key purposes here are related to control the type 1 error rate, maintain integrity of study results, ensure valid and reliable conclusions and support regulatory compliance. Now the definition here is multiplicity analysis controls increased false positive risk when multiple statistical tests are performed. Now why multiplicity matters? Now suppose there are 20 endpoints tested. The alpha level of significance is 0.05. By chance alone, some endpoints may appear significant. So what happens is this inflates the type 1 error, which is the false positive. To explain in a better way, say I have a real world example is studies test which OS is overall survival, PFS is progression free survival, ORR is odds response rate, then QOL, which is quality of life, biomarkers and several doses. So these are the study tests that is being used. Now without adjustment, false positive probability increases substantially. Now say for example the level of significance alpha is 0.05. There are 3 endpoints, OS, PFS and ORR. Now according to the Bonferroni method, if there are 3 endpoints which I am going to do the multiplicity analysis, so the level of significance will be divided by 3 because I am having the 3 endpoints, right? So each endpoint must achieve P which is less than 0.0167. That comes from dividing 0.05 divided by 3. So that's how you need to check the multiplicity analysis if it is 3. If there are 4 endpoints, then alpha level of significance will be divided by 4 and whichever that p value comes, that p value should be less than to that number. Now the hierarchical testing examples test overall survival first. If significant, then you go to the next level, which is PFS. If PFS is significant, then you go to the next level and test ORR. Now what happens if the overall survival fails, which is the primary endpoint, right? So if the OS fails, testing sequence stops. Remaining endpoints considered descriptive. Descriptive is nothing but summarizing only the summary stats, like say for example mean, median, then what is the minimum, maximum, range. So these are all descriptive statistics that you present. You don't conclude any analysis for other parameters once the primary endpoint fails. Now, what is the relationship between clinical trial endpoints? Now, each endpoint type plays a distinct role together, building robust and reliable clinical evidence. Right? Now, whenever used appropriately, these endpoint types work in harmony to generate meaningful and impactful clinical evidence, because you also need the USA FDA approval, to launch your molecule or a medicine into the market. So the relationship between these is, like, say, the primary endpoint, the main purpose is to main objective of the study, and the regulatory importance is very high. So this is considered as the most important. Now remember one thing, if the primary endpoint it fails, like, to prove the significance, then for the remaining endpoints you just show the descriptive statistics and you just submit that clinical trial study. It doesn't make sense because the primary objective, it has failed the safety and efficacy parameter for that clinical trial study. If it passes the primary endpoint, then you can go ahead for the remaining endpoints. Now the secondary endpoint here is additional benefits is the purpose, and the regulatory importance is high. Exploratory is hypothesis generation, and it is moderate to low importance. Sensitivity is robustness validation, and it is very high importance. Multiplicity is control false positives, and it's very high importance in regulatory importance. End-to-end clinical trial example. Here is an oncology phase 3 clinical trial where the primary endpoint is OS, secondary endpoint is PFS, ORR, and QOL. The exploratory endpoints are related to biomarker subgroup changes, ctDNA changes. Sensitivity analysis is related to different missing data assumptions and per protocol analysis. And multiplicity control is hierarchical testing sequence. Now what is this? Like I explained earlier, if OS is significant, then you go for PFS. If PFS is significant, you go to ORR. If ORR is significant, you go to QOL. So this is the testing sequence that comes under the multiplicity control. So you're controlling each and every endpoint, and then you're going ahead step by step. Now, clinical SAS programmers support endpoint analysis by primary, which is related to TLF generation and survival analysis SAS activities. Secondary area is related to additional efficacy tables. Exploratory subgroup outputs, which I explained in the above slides, related to the HIV and AIDS related kind of a clinical trial. Sensitivity is reanalysis, like say you submitted the analysis based on including missing data. So the reanalysis, it could be you use the LOCF or you use some other imputation methods and regenerate the analysis data sets and regenerate the TLF. So that comes under the sensitivity analysis. And multiplicity, like I explained in earlier slide, it's a p-value adjustment programming. Now it's a very simple in a layman's common language that I will explain here is clinical trial, the primary endpoint is nothing but the final exam score, right? Secondary endpoint is nothing but an assignment scores, right? Exploratory endpoint is nothing but the creativity assessment. Sensitivity analysis is nothing but rechecking grading fairness, right? And multiplicity is avoiding too many bonus tests causing unfair pass. So this is one example which I am trying to explain in a very common layman's language, where you are imagining evaluating a specific student. So here I have just tried to create an analogy. Now the summary here is the analysis type related to the main goal of these endpoints. Now the primary is to prove the treatment works, right? Secondary is to show additional benefits. Exploratory is to discover the future insights, whether I would like to conduct the clinical trial study if I try to test this specific hypothesis generation in this study, and based on the sufficient or satisfactory results, if I can discover another clinical trial or for conducting the future clinical trial study. So that is the exploratory. Sensitivity is verify robustness for including missing data or using the imputation methods. Multiplicity is controlling your alpha rates, which is the preventing false positives. That means you're controlling your type 1 error rate. Many thanks for watching my video and happy learning. Please like, share, subscribe leave a feedback comment. Many thanks for your support and having great connection.